In this video, I want to take you through some of the ways that bacteria, prokaryotes, control transcription and translation. Specifically, I'll cover the crucial steps of initiation and termination of transcription and translation, which are the key regulatory steps that control protein production. We'll also look in detail at the specific nucleotide sequences that control these steps, hopefully so that we can identify them in nucleotide sequence. And then I'll finish by showing you how changes to these regulatory sequences, so specific DNA mutations, would actually affect protein production. So the central dogma of molecular biology states that all the information for making proteins is stored in DNA, and that information firstly gets transferred to RNA in a step called transcription. And then the sequence information in the synthesized RNA molecule is used to determine the order of amino acids in the final protein in a step called translation. Now, while outside the central dogma itself, but still key to the regulated production of proteins, the polypeptide chain that's formed must then fold into the final active protein. Sometimes this folding step happens unaided, sometimes other proteins known as chaperones might be involved to catalyse the correct folding, and sometimes multiple polypeptides may come together in this step. Now, while the central dogma as shown is the normal order of steps in the flow of information, we know that information can sometimes flow from RNA back to DNA via the action of reverse transcriptase. But we're not going to really focus on this step in this video. What I do want to focus on, though, is what sections of the DNA sequence are critical for initiation and termination of both transcription and translation. So we want to know precisely where along a DNA molecule does transcription start and finish, and also where does translation start and finish along the RNA molecule. Importantly, I want to discuss how we can identify the particular nucleotide sequences that control these steps of initiation and termination, and discuss what these sequences can actually tell us about when and in what quantities proteins are made. So let's look first at transcription, as that's the first step. So transcription is the first step in expression of a gene and is an absolute prerequisite for protein production. If a gene is not transcribed, then its corresponding protein can't be made. So transcription is the copying of a segment of DNA into RNA, which is driven by the protein called RNA polymerase, or perhaps more specifically, DNA-dependent RNA polymerase, or sometimes shortened just to RNAP. So for transcription to occur, RNA polymerase must associate with the DNA, melt or unwind the two strands, and then synthesize an RNA copy of just one of the DNA strands. So in the image shown here, the bottom strand of the DNA is being used as the template strand. So the new RNA that's being synthesized, which is shown here in red, will be complementary to this sequence. The top strand of the DNA in this region is called the coding strand, and it's the same as the newly synthesized RNA, except of course that RNA contains uracil in place of thymine bases that are used in DNA. So the newly synthesized RNA is always made in the 5' to 3' direction. And so the RNA is extending out in this direction here, and the RNA polymerase is also moving in that direction. So as RNA polymerase proceeds here, it will make the RNA strand and then move the unwound bubble of DNA further to the right in this diagram as it continues to synthesize the new RNA. Now, bacteria have a single RNA polymerase which transcribes all the genes. The active bacterial RNA polymerase has 6 subunit components. It has 2 alpha components, a beta prime, an omega, and a sigma. And we'll look at those components in a couple of slides. Now, I don't want to focus on eukaryotic transcription and translation in this video, but as a comparison, eukaryotes have 3 different RNA polymerases, designated 1, 2, and 3. And each of these types of RNA polymerases transcribe a different set of genes, with RNA polymerase type 2 transcribing all of the protein encoding genes. Now, the eukaryotic RNA polymerases have similar core components to bacterial RNA polymerase, but often have other subunits as well. So given that transcription is driven by RNA polymerase, then initiation of transcription must be determined by where RNA polymerase first binds. So transcription starts when RNA polymerase binds. And the position where this happens is called the promoter sequence. On the other hand, termination of transcription must be where the RNA polymerase dissociates from or falls off the DNA. And this happens at the transcriptional terminator. So what are the specific DNA sequences that define promoters and transcription terminators? How would we find them in a piece of DNA like the one shown here? Okay, so let's start with promoters. So bacterial promoters are defined by 2 short sequences that interact with, or actually bind to, RNA polymerase. And also the same regions must allow RNA polymerase to unwind the DNA at that local region. So the strands can come apart and RNA polymerase can have access to a single strand so that it can make the RNA copy. So a common promoter sequence has a 6 nucleotide element called the -10 region and a 6 nucleotide element called the -35 region. These are the specific regions where RNA polymerase binds, and these -10 and -35 sequences are critical for the interaction of the DNA with RNA polymerase. As well as being the area where RNA polymerase binds, also this -10 region is highly AT-rich, which allows RNA polymerase to easily separate the strands of DNA at this region. This is where the melting actually occurs. Now, the -10 and -35 regions are named as they are that many bases upstream of where transcription actually starts. So these -10 and 35 regions are not actually themselves transcribed. But in fact, the first base that's transcribed is designated +1. And this, in this instance, is the C here. So the +1 is 10 nucleotides downstream of the middle of the -10 element. So we could count it like this. And we would see that there were 10 bases in between the middle of that element and where transcription actually starts. So now the promoter sequence shown here is actually the consensus sequence for the normal E. coli housekeeping type promoters. And that sequence is this TTG-ACA at the -35 position and TAT-AAT at the -10 position. Now, not all promoters are exactly the same. Indeed, here's quite a large number of actual sequences of different standard housekeeping E. coli promoters. For each of the genes, each in a different row here, the promoter sequence upstream of that gene is shown, and highlighted in red are the common bases across all of those sequences, and the -10 element and the -35 element are shown here under these boxes. So in fact, you can see here that very few of these promoters actually have exactly the same sequence as the one I showed you and called the consensus on the previous slide. So you see very few of them actually have TTG-ACA at this -35 site. There's one here, the TAC promoter. And similarly at the -10 element, very few have TAT-AAT. There's usually 1 or 2 bases different, but again, the TAC promoter has a perfect TAT-AAT consensus here. But the most commonly used bases are indeed those consensus bases that I showed you, that TTGACA and TATAAT. Now, the fact that these different promoters can have different sequences is an important first step in the regulation of transcription. So RNA polymerase will bind with different efficiency to these different sequences. It'll bind more strongly to some and less strongly to others. In turn, this will affect how often the promoter binds RNA polymerase, and therefore how often the downstream gene is transcribed. So in general, if a gene is preceded by a promoter with a sequence that's close to the consensus, it'll be transcribed more often and therefore likely produce more protein than a gene that's preceded by a promoter that's less similar to the consensus, as it won't bind RNA polymerase as efficiently, so won't bind it as often, and so won't be transcribed as often. So as I said before, RNA polymerase is a multi-subunit enzyme. It has 6 subunits. So the RNA polymerase core enzyme consists of 5 subunits. So here's the core enzyme up here. There's 5 subunits, 2 alpha subunits, beta, beta prime, and omega subunit coming together. So the core is actually catalytically active, but can't bind to specific DNA sequences. It can't bind to the promoter sequence. So specific binding to promoters is driven by the sigma subunit, which is sometimes called sigma factor. So the sigma subunit specifically binds to the -10 and -35 sequences. So it actually recognizes the specific DNA sequences of the promoter. So it comes and binds here, recognizing those 2 6 nucleotide sequences at the -10 and -35. Once it's bound, the rest of the core comes and interacts with sigma to give the fully active 6 subunit, what we call holoenzyme. So the binding of RNA polymerase is the most crucial regulatory point in protein production. If RNA polymerase can't bind, then the gene won't be transcribed and the protein won't be made. Alternatively, if RNA polymerase binds more easily, then more transcript will be made and likely more protein will also be made. So much of gene expression control is centered around controlling transcription initiation via this binding of RNA polymerase. So repressor proteins are important regulatory proteins that regulate transcription initiation. Now, repressors generally bind in a position that will stop RNA polymerase sigma factor binding and or DNA unwinding by RNA polymerase. For example, the LAC repressor protein LAC-I binds between the -10 and the +1 nucleotide, stopping initiation of transcription. So you can imagine the LAC-I proteins bind in this region here. And under that situation, the sigma factor can't bind with the -10 region here, so is never able to bind. RNA polymerase doesn't come together here, and so transcription doesn't start. So these repressors inhibit transcription initiation by stopping RNA polymerase binding. Alternatively, activator proteins stimulate either RNA polymerase binding or DNA unwinding. So an example of an activator that facilitates RNA polymerase binding is Fizz. Now, Fizz binds upstream of the -35 region. So here. And here it's in a position where it can interact with the alpha subunit of RNA polymerase. So after sigma factor comes in, and then the rest of the complex comes in to form the final holoenzyme, we can actually get an interaction here between Fizz and the alpha subunit on RNA polymerase that actually facilitates and stabilizes the binding of RNA polymerase with the DNA. So in general, repressors are going to stop RNA polymerase binding, and activators are going to increase the likelihood of RNA polymerase binding. Now remember that the sigma factor controls the specificity of RNA polymerase binding. Now, under certain conditions, cells can express different types of sigma factors. So the most common sigma factor in E. coli, and the one that's used to express most of the common housekeeping genes, is sigma 70. But other sigma factors are also made, and these recognise different sequences. So here's a table showing some of the different sigma factors that E. coli can make. We see the sigma 70 standard sigma factor that I showed you before. It has the TTGAC minus 35 region and the TAT-AAT minus 10 region. But there's a number of other sigma factors, and they recognise different sequences. This allows the cell to rapidly swap to expressing different types of genes which have promoters recognised by the different sigma factor. So for example, under heat shock, bacteria express the heat shock sigma factor, which is the one shown here. This sigma factor recognises promoter sequences like TTG-AAA and 4Cs AT and T. So because this sigma factor recognises different sequences, when this is switched in, this results in hundreds of new genes being expressed, as these new genes have promoters that are recognised by this new sigma factor that wouldn't have been recognised before by the sigma 70 sigma factor. So switching of sigma factors, again, is a crucial regulatory point for protein production in bacteria. So now let's return to our normal sigma 70 promoter with bound RNA polymerase. So once bound, the polymerase now melts or unwinds the DNA at the minus 10 region and begins transcription from the +1 nucleotide. Note that the RNA polymerase has a structure which allows specific DNA entry, unwinding and exit. So the DNA is going to come in through this section here. It's going to exit here. As it comes in through the entry channel, the strands are melted, they come apart, and then you can synthesize the new RNA that's complementary to this template strand here. And there's also a channel here for the ribo NTPs to be able to enter, which are necessary obviously to make the newly growing RNA strand. So now here we see a schematic of the situation after RNA polymerase has unwound the DNA, beginning at the minus 10 TAT-AAT region. That's shown in the sequence just here, so it's our TAT-AAT through here. So the unwinding starts there, and now we have an unwound bubble where transcription can actually start. So with this free single strand at the bottom here, which can be used as a template to make the new RNA. Remember that the RNA synthesis occurs in the 5' to 3' direction, so it's going to be going in this direction here. And that the RNA polymerase, which is the one shown in brown here, the complex, is going to also move here, and so is going to continually expose new nucleotides for RNA synthesis as it moves. So here we see the situation just after the first few bases have been transcribed into the new RNA molecule. And as that continues, here we see the situation after the RNA polymerase has moved further along the DNA. As the RNA polymerase continues to travel along the DNA, the newly synthesized RNA comes away from the DNA as the DNA molecule rewinds. So here we see the DNA has gone back to double-stranded as it exits the RNA polymerase, and the newly formed RNA molecule comes out here. So RNA polymerase will continue to transcribe the RNA so long as it stays in contact with the DNA. So what's actually going to stop transcription? What's going to make that RNA polymerase fall off and stop making the RNA transcript? So transcription is terminated by sequences called transcription terminators. In bacteria, there are two different types, row-independent and row-dependent. So row-independent terminators, often called intrinsic terminators, just require a termination sequence on the DNA. So that sequence is a short palindromic GC-rich sequence that's followed by a run of Ts. Row-dependent termination, on the other hand, requires the action of a protein called Rho. These Rho-dependent terminators have less well-conserved sequence components. So let's first look at Rho-independent terminators, which are probably the most common in bacteria. So what does this Rho-independent terminator sequence look like? So as I said, it must have a short, usually GC-rich palindromic sequence, which is followed by a run of Ts. So first of all, what's a palindrome? So a palindrome is a sequence that reads the same from the 5' to 3' direction on one strand as it does 5' to 3' on the other. So if we look carefully at this sequence, we see this region here on the top strand reads GGC, GCG. And on the other strand, going the other direction, we also see that it reads GGC, GCG. So that's the start of a palindrome. The end is also a palindrome. So here we have CGC, GCC, CGC, GCC. So these outside sections of this sequence are palindromic, but the 4 bases in the middle are not palindromic. So in general, terminator sequences have palindromic ends, and the central few bases are not part of the palindrome. So here, this one would be the palindromic sequence is the GG, CG, CG, gap of 4 nucleotides, CG, CG, CC. So when this region of DNA is transcribed, we'll make an RNA copy of the bottom strand here. Now, while this sequence is being transcribed, because RNA is single-stranded, the RNA molecule can actually pair with itself at the palindromic sequences to create what's called a stem loop. So the exactly complementary bases form the bound stem. So we can see here, this is the sequence of this RNA. We can see first of all the ACU, AAUC, that's this bit here, ACU, AAUC. Then we go through the A's, and then we get to the bit that's part of the palindrome, this GG, CG, CG. And because that's a palindrome, it's the reverse complement of this sequence down here, which means that those two can bind to each other to form this stem. So this first G combined to this last C, which is the one shown here, the next G bind to this C, etc. Now, as I said, the 4 bases in the middle aren't part of a palindrome, so they don't pair with each other and they form this unbound loop up the top. So this is called a stem loop, and you can see why, because it has this nice stem with bound nucleotides and then a loop of unbound nucleotides at the top. So it's this ability to form the stem loop that's critical for the transcription terminator. We'll also see that the run of U's downstream of this loop is also important. So if we look now at schematic of how the RNA in this region would actually be transcribed by the RNA polymerase, we can see that the stem loop can form as the RNA is being synthesized. So here I'm using exactly the same sequence as shown in the last slide. I know it's a bit hard to see, but you can stop it and take a look and compare the sequence that I had on the last slide with the one here. So as the RNA is being synthesized, this RNA as it comes out here can form this stem, those pairings that I showed you before. And this formation, the formation of this stem loop here, causes the RNA polymerase to pause. So it stops moving forward. Now when the stem loop's completely formed, and in this instance I've still got 2 more bases to form the full stem loop. So once that's completely formed, the only RNA-DNA interaction here will be between the A's and the U's. So these are very weak pairings. So the AU pairing, just 2 hydrogen bonds, a long run of A's and a long run of U's gives a very weak pair. And while the RNA polymerase is stalled, paused at this region because of the stem loop, only being held together with the DNA by these very weakly interacting AU pairs, then RNA polymerase dissociates from the DNA, falls off, and this terminates transcription. So that's the most common way probably that transcription terminates, because for transcriptional terminator that has a sequence that has a GC-rich stem loop palindromic sequence followed by a run of Ts. They're actually pretty easy to identify in nucleotide sequence, and we can often predict where such terminators are. So the second type of transcription terminator is a row-dependent termination. And as I said, this requires the action of a protein, the row protein. Now this type of termination has much more poorly defined sequence elements, so I'm not going to show any specific bases. But again, I'm going to show you a schematic of RNA polymerase moving on the DNA and synthesizing the new RNA strand. So here it is the same as before, but I'm just not showing you bases because the bases, while there are some bases which are likely important, we don't really know the sequence specificity very well. Now, as I said, for termination to occur, we need the row protein. So the row protein is a hexameric RNA helicase. Here's a 3D structure. And this RNA helicase is able to bind to RNA and move along the RNA using energy from ATP hydrolysis. So in schematic form, the row protein looks something like this. So the row protein actually binds to the newly synthesized RNA transcript. So it's going to bind somewhere along this RNA molecule here. There's actually a sequence that row specifically binds to, and that's called the row utilization site. Or RUT. And once row binds to that site, then it moves along the RNA. So row first binds to that site, sitting on the RNA transcript, the newly synthesized RNA transcript, and then it begins to move along the RNA. So the binding site is usually about 70 nucleotides in length, and it's C-rich and G-poor, and that's pretty much all of the common sequence components that we know about. Now row moves along until it hits the RNA polymerase. Now usually this only happens after the RNA polymerase pauses due to some specific sequence on the DNA template. Then once row catches up with the RNA polymerase, it kicks the RNA polymerase off the DNA, causing it to dissociate. So in summary for this section, transcription is the first critical step in expression of a gene. Transcription begins at the promoter sequence. This is where RNA polymerase binds to the DNA and usually is a set of hexameric sequences called the -10 and -35. And these are often like TTG, ACA as the -35 and TAT, AAT as the -10. So RNA polymerase binding to the promoter is dependent on the sigma factor, and regulatory proteins such as repressors and activators often act to alter the efficiency of binding of RNA polymerase to DNA, and in this way regulate expression of the gene that follows the promoter. Transcription ends at a transcription terminator. So this is where the RNA polymerase dissociates or falls off from the DNA. So we have two types of termination signals in bacteria. Row-independent termination or intrinsic termination is at palindromic sites that can form a stem loop and are followed by a run of Ts. And row-dependent termination requires the presence and action of the row protein and occurs at less well-defined sequences. So we've covered initiation and termination of transcription, and know that transcription is going to start at a promoter site. -10 and a -35 region here is going to define that promoter. Transcription is going to start there and then it'll continue until it comes to the terminator site, somewhere further down on the DNA. So this generates an RNA of a particular length, And the next step is translation, which will turn the sequence of bases in this RNA into the sequence of amino acids to give our protein. So here's the 5' end of a possible messenger RNA. It's the same sequence that we started with earlier in the video when we were looking at transcription initiation. So how's this going to be translated? Where's translation going to start? Where's translation going to finish? So translation is performed by the ribosome, which is made up of two subunits, the large subunit, or 50S, and the small subunit, or 30S. So for translation to happen, the ribosome has to dock onto the correct position on the RNA. So it's got to find the spot where it needs to start its translation. And then after it finds that spot, it'll move along the RNA, translating each set of 3 bases, each codon, into the appropriate sequence of amino acids to make the protein. Now, how does the ribosome know where to bind on this piece of RNA? So obviously, the start codon is going to be important. So usually bacteria use a methionine AUG codon as the start codon. So that's AUG on the RNA, that would be ATG in the original DNA. So AUG start codons actually are used in approximately 83% of all the genes in E. coli at least. But there's a couple of other start codons that can be used. So GUG is used about 14% of the time, and UUG is used 3% of the time. So in the example shown up here, the RNA does in fact have an AUG start codon, and that's shown here. But the ribosome doesn't just recognize the start codon, because there'd actually be many AUG codons spaced through this region. There's not actually another one shown in this small piece of RNA, but along this piece of RNA that's encodes some protein, there'd probably be tens or more AUG codons. So how does the ribosome find the right one and know to place itself right in front of that starting first AUG codon? So in fact, the ribosome places itself onto the RNA at a sequence at the start of the RNA called the ribosome binding site, for obvious reasons, or the Shine-Delgado sequence. And Shine-Delgado were the two people who first proposed that this sequence was where the ribosome docked onto the messenger RNA. So that ribosome binding site normally is something like GGA-GG. And we can see that sequence here. So this is the ribosome binding site, and this is where the ribosome is going to dock. And we'll see that that sequence is actually just upstream of the start codon that the ribosome will want to begin translation from. So how does the ribosome find this ribosome binding site or Shine-Delgado sequence on the messenger RNA? So actually, the 30S subunit of the ribosome has an RNA component called the 16S ribosomal RNA. So 30S subunit's got proteins plus this ribosomal RNA subunit. And a section of that RNA is CCUCC, as shown here, which is the exact complement of the ribosome binding site, which I showed you before, this GGA-GG. So in fact, that's how the ribosome finds the correct spot. So it actually binds exactly to that ribosome binding site sequence. And when it binds at that point, it puts the ribosome in just the right position so that the start codon, which is between 4 and about 7 or 8 base pairs downstream of that GGA-GG, it puts that in exactly the right position. next to this site where translation is actually going to happen. So here we see the RNA sequence written out again in freehand with the ribosome binding site now shown exactly opposite and bound to the section of the 16S ribosomal RNA that is complementary to it. And as I said, this allows the messenger RNA to be placed precisely so that the AUG is under this P site. Now, the ribosome has these three sites, E site, P site, and A site, as written here. So the A site is the aminoacyl tRNA site, the P site is the peptidyl tRNA site, and the E site is the exit site. So new aminoacyl tRNAs normally come into the A site, except for the first tRNA. The first tRNA, which is F-met, and it comes into the P site, which is situated right above where that AUG start codon is. So in the cell, there'll be tRNA molecules floating around, and one of those tRNA molecules will be carrying the F-met amino acid on there, and it'll have an anticodon, which is exactly the opposite of AUG. So this would have UAC, and this would then come in and bind here in the P site to the AUG that's here. So this is the start of translation and the start of protein synthesis. So once that F-met tRNA is in place, then the next codon has been placed directly under the A site. And so the tRNA with an anticodon that is the exact complement of the three bases in this next A site, which in this case would be the histidine tRNA, which would have a GUG. This will now come in and fill the A site. When that happens, we now have these two tRNAs placed appropriately, and the ribosome can make a bond between these two amino acids here. So once this peptide bond has been formed between the two adjacent amino acids, then the 50S subunit moves along to the next codon. It also moves these two tRNAs so that they are now in the E and the P site rather than the P and the A site. Once the F-met tRNA enters the E site, that's the exit site, that will then leave, and we'll have a new space for the next amino acid in line to come in. So this continues over and over with the ribosome translocating along the messenger RNA, adding the next tRNA, making the new peptide bond each time, and synthesizing the protein. And this happens until the A site becomes placed over a codon that has no complementary tRNA. This is what we call a stop codon. And so the stop codons are UAA, which is the one shown here, but also UAG and UGA. So these are the three stop codons that they have no complementary tRNA, so they don't specify any amino acids. So once this codon is in place at the A site, then there's no tRNA that can come in. When this happens, the ribosome dissociates from the messenger RNA and releases the synthesized polypeptide. So in summary for this section on translation, translation is performed by the ribosome, and it begins at a start codon. Usually an AUG, but there are two other possibilities as well. However, the ribosome has to place itself in the right position, not based on the start codon, but on the ribosome binding site. So the ribosome binding site, or Shine-Delgado sequence, usually something like GGAGG, is going to guide the ribosome to the correct spot so that the AUG start codon is just in the right place beneath the P site. And of course, the ribosome recognises that ribosome binding site via complementarity to the 16S ribosomal RNA. Then translation ends at the stop codon. So it ends when there's a codon brought into place that there's no tRNA that recognises that codon. And so the A site becomes empty and the ribosome falls off and releases the completed protein. Okay, so now I want to look at what happens during transcription and translation on a larger scale. So remember, in prokaryotes, there are no membrane-bound organelles. So transcription and translation happen in the same space, so in the cytoplasm of the cell. So this means that they can happen concurrently. So here I'm showing a piece of DNA with a promoter, and you can see the -10 and the -35 regions on this promoter. And I'm not showing a terminator on this piece of sequence, which means that we have a starting point for transcription, but no stopping point for transcription, just in the piece of DNA that I've shown on the screen here. So what's going to happen? So because there's a promoter here, the RNA polymerase, which I'm showing in yellow here, will be able to recognize this promoter sequence, remember, via the sigma factor, and begin to move along here and transcribe the RNA. So as it continues, the newly synthesized RNA, which is shown in red, will start to appear out from the back of the RNA polymerase. And those sequences which allow translation to start, so remember our ribosome binding site, which is our GGAGG, and our start codon, which is probably AUG, these will appear, and they'll be free in the cytoplasm to now interact with the ribosome and for translation to start. So as transcription continues, and we see now the RNA polymerase moving along, and the messenger RNA that it's synthesizing getting longer. Now the ribosome has found the ribosome binding site and the start codon, and is now going to begin to translate this message while transcription is still happening. And another RNA polymerase may also bind and begin a new round of transcription. So we can have multiple rounds of transcription and translation all happening at the same time. And as this continues even further, we can now see that we have two different transcription events happening. The first one has now been transcribing for some time. The ribosomes have hopped on. They've started to translate and make the protein, which is shown in green. This one's obviously done more translation than this one here, but this one's also starting. And as these both move along, then probably another ribosome will come and start to translate this again. And we also have translation happening now on the next transcript, which is also being made by this RNA polymerase, which is moving along the DNA. And so we can actually see this in an electron micrograph. And so here's one showing both DNA, RNA, and the ribosomes. And so we see the DNA here, which is being transcribed by multiple RNA polymerases to make messenger RNA. So here's one of the longer ones. Looks like RNA transcriptions proceeding in this direction, one of the longer ones, and some shorter ones through here. And each of these messenger RNAs, just like the ones shown here, have ribosomes translating them many times. So we have multiple ribosomes on each messenger RNA making the proteins. So in the last section of this video, now that we've covered the sequences of the elements that control initiation and termination of transcription and translation, I want to look at where we'd expect to find these elements on a whole genome scale and how changes to these sequences would affect protein production. So in the examples I used earlier for transcription, I showed the top strand as the coding strand and transcription moving from left to right. So top strand, coding strand, RNA polymerase moving in that direction. So in this instance, the promoter would have to proceed where transcription starts, so the promoter would have to be down this end, and the terminator would be at the end to stop transcription, so it would be somewhere down here. I'll use these right-angled arrows and these stem loop symbols to designate promoters and transcriptional terminators as I go through the video. However, it could just as easily be that the coding strand is on the bottom. It would just mean that we're looking things around the other way. And so in this case, if the coding strand is now at the bottom, then that means that transcription is going to move from right to left. And the promoter then would have to be at this end, and the terminator would have to be down this end. Now, transcription moving in this direction still makes perfect sense because, of course, the strands of DNA are anti-parallel, which means that this is the 5' end of the bottom strand and the 3' end of the bottom strand. So the direction of transcription is still 5' to 3', and our new RNA is being made 5' to 3' as it must be. Of course, in reality, on a whole genome, some genes are encoded on one strand and some genes are encoded on the other. So if I show a larger region of DNA and the two strands, top strand 5' to 3' that direction, and the bottom strand 5' to 3' in the other direction, then some genes will have their coding strand on the top strand. On the other hand, other genes will have their coding strands on the bottom strand, which means that transcription will move in different directions at different places around the genome. In each case, it's the coding strand for the gene that allows us to predict where the other elements will be. So the standard way of presenting this for large sections of a genome is to show each gene with an arrow indicating which direction it's transcribed. So for the ones on the bottom strand, they'll be being transcribed in this direction. For the ones on the top strand, in this direction. So here we have a representation of about 16 kilobases of a genome. And this section has 8 genes. As you can see, the arrows go in different directions, indicating that some genes are going to be transcribed from left to right, and some genes are going to be transcribed from right to left. So if the gene is transcribed from left to right, like all of the genes here in red, then the coding strand for that gene must be on the top strand, just as we showed earlier for those initial schematics on transcription. So the gene's being transcribed left to right, as all the red genes will be, then that means that the promoter sequences have to precede the gene. So for gene B, a promoter down here, and at least another promoter down here. It also means, of course, that the transcriptional terminator sequences have to be after the gene. So for gene B, one in this region here, and E, F, and G may be terminator down here perhaps, but we'll talk about that in a bit more detail in a minute. Alternatively, the genes in green are transcribed from right to left, so must have their coding strands on the bottom strand. So their promoters will then have to lie to the right, and so we'd find them in these regions here. Now the promoter element sequences would also have to be on the bottom strand and read from the 5' to 3' direction. So for this promoter here, we'd expect the -10 and the -35 to be on the bottom strand and reading 5' to 3' on the bottom strand, TTG, ACA, or something similar to that, and TAT, AAT, or something similar to that. But they're going to be on the bottom strand, reading in that direction. So now I want to focus just on these genes in this region here. So if these genes are real and transcribed, at least under some conditions, then there must at least be a promoter here, going in this direction, and a promoter here, going in this direction. So the top promoter is going to transcribe from the left to the right and is therefore going to transcribe gene E. The promoter shown on the bottom is going to transcribe right to left and at least transcribe D. Now, where would we expect the terminators to be? Well, there could be a terminator after gene E, which would mean that we'd get transcription beginning at the promoter in front of E and just going to the end of E and then stopping. If this was the case, then we'd need another promoter after this here to drive transcription of gene F. But bacteria, and only bacteria, can have genes transcribed together in what we call operons. So an operon is a set of genes that are transcribed from the same promoter. So it's quite possible that these three genes shown in red could all be transcribed as a single operon, in which case there'll just be the one promoter, the one I've shown here, in front of E, and one transcription terminator following G. And they'll all be transcribed together on the same mRNA message, which will run all the way through there. Now, how about this set of two genes on the left? So there will definitely be a promoter in front of D, the one I've already shown here. But again, they could be transcribed as a single operon or as two separate genes. So for the sake of this video, let's say that these ones aren't an operon. So this section has two promoters, one in front of D, one in front of C, and two terminators, one at the end here and one at the end here. So we get two transcripts, one going through here and one going through here. So let's look in more detail at these three genes. So we've said they're an operon, so that means that we have a promoter down this end, a terminator right after all three, and no terminators in between. So there's going to be transcription starting at the promoter and running all the way through until we hit the terminator. So on that single messenger RNA, we're going to have the coding sequences for these three different genes. So that means that upstream of each of those genes, there'll likely be a ribosome binding site. Now, ribosome binding sites aren't always exactly the same. Some are a little bit more efficient than others. So perhaps this one just has GAG up in front of it rather than the more efficient GGAGG. And they'll each have their own start codon. So right at the start here will be an AUG, and here, and here. And they'll each have their own stop codon. showing the 3 different types of stop codons that you can have. So as these coding sequences each have their own ribosome binding site and start codon, they can all be translated separately, and they will be. So the ribosome can come and bind to this first ribosome binding site, placing its P site right at that start codon there. Another one can come and bind here. Another one can come and bind here. And then they'll start to translate each of these coding sequences separately. So for example, the middle one, the ribosome, will move along here, making the protein until it gets to that stop codon, and then it'll fall off. Of course, we can actually have multiple ribosomes. So one might start here and start to translate along here, and when it's at this point, another ribosome might come on, and so we'll have multiple proteins being made at the same time from the one message. And of course, this can happen for all 3 of the coding sequences. So finally, let's look at a couple of possibilities for what might happen if there were mutations in this region of the genome. So what about if during replication, randomly there was a mutation that happened at this run of GGAGGs here that form the ribosome binding site of this second coding sequence? So perhaps this sequence was mutated to be completely deleted. So this ribosome binding site sequence is now absent. Everything else is still intact. We still have our promoter sequence up here. We still have our terminator sequence down the end. So we're going to get transcription. There's been no change to transcription. This change in the middle here makes no difference to transcription. It's going to start at the promoter, go all the way through, and finish down here. We still have all of the coding sequences intact as well. The AUG here, stop codon, start codon, stop codon, start codon, stop codon. But the mutation has taken out this ribosome binding site just here. No effect on this one, no effect on this one. So what do you think is going to happen? So the ribosome can still bind here and translate this gene. It can still bind here and translate this gene. But it can't find where to go here. There's no ribosome binding site. So it never jumps on there. So we'll only ever get proteins made from this gene and from this gene. And they'll come from these ribosomes that combine, ribosome binding site, move through and translate these 2. So the mutation to the ribosome binding site in front of gene F means that we actually get no protein F. Made. Even though it'll be transcribed, it won't be translated. On the other hand, what would happen if we had a mutation to one of the start codons? So let's say the start codon for the first gene was mutated. So this sequence in here, rather than being the AUG, which is the normal and required start codon, maybe we had a mutation which takes that to AGG. Okay, so if this sequence is now transcribed, which it will be because we haven't changed the promoter sequence, we haven't changed the terminator sequence, so we'll still get transcript made exactly the same here, except just this one base here in the middle is changed. But now we have AGG instead of AUG. So what's going to happen under this circumstance? Well, there's no change here. Ribosome, still got a ribosome binding site, can still bind and translate and make protein G. Ribosome can still bind here. The ribosome binding site, gone back to the natural one here now, and start codon here, starts there, moves down, translates. But even though the ribosome can recognize this ribosome binding site, there's now no longer a start codon right at the correct position after the ribosome binding site to allow translation to begin. So this gene E is never going to be turned into protein. So again, we'll be able to get protein made of gene F, and we'll be able to get protein made from gene G, but we won't get any protein made from gene E in this situation. Now, what would happen if we had a mutation to the promoter region in front of E? So I've said there's a promoter in front of gene E, so that must mean that there's some -10 and -35 element, and say that this is a consensus E. coli -10 and a consensus E. coli -35. So this is the sequence in this region here. But what about if we had a mutation that changed the -10 region? So what if that sequence was mutated, so now it read T, G, G, C, C, T. So this is a very poor -10, almost certainly won't work as a promoter at all because it's no longer AT rich, it won't bind the sigma factor properly, and even if there was some binding, it basically wouldn't be able to unwind this section of DNA at that region. So with this mutation here, this promoter is not going to work. So now just these few bases changed in this region up here in front of the gene will mean that we get no transcript made of any of these genes. So that means that we get no transcript for all three, so no protein. So a small mutation, but just in the right place up here, means that we lose the ability to make all three of those proteins.